Okay, this is chapter 19 where we're talking about blood vessels. Let's jump right into it. Before I start, I want to remind you, you have a list of all the vessels that you need to know, both the coronary vessels and outside of that, the veins you need to know, the arteries you need to know. You've already been provided with those lists. You're going to be tested on them very quickly. You need to start studying them now if you haven't been already. I hope you've been doing it before. Anyway, let's jump into this. All right, so why do we need to know about blood vessels? Because blood vessels deal with blood pressure, and we're going to talk about how to do blood pressure. Anyway, let's do the little video. Okay, let's move on. So blood vessels are says a delivery system of dynamic structures that begins and ends at the heart. So let's break that down a little. Dynamic, that means it's not static. It's not just a pipe. These things, they have elastin in them that can allow some of them to get really swelled big, like the aorta gets, and then it returns to its normal size. That helps to dampen the pressure changes that you would have if you just had the heart. You'd have if you just had the heart dealing with pumping, the swings in pressure between high and low, beating, pumping, and resting, they would be much bigger changes. But these vessels can change the pressure, reduce it, spread it out a little, make the pulse a little. You still have the same extremes, but spread over a longer period of time, and slow it down. So that's good. But also getting bigger, getting smaller to control the overall pressure in the system, control where blood flows. If you're running from a bear, you're not going to be sending a lot of blood to the stomach at the moment to do digestion. That's not necessary. Um, Arteries always carry blood away from the heart. Veins always carry blood very close to, I should say, towards the heart. Veins always carry it to the heart, arteries away. Um, Capillaries are the very, very thin walled vessels, tiny vessels. They go through the tissue. And some of them are so narrow that red blood cells have to turn sideways and go single file down them. Anyway, but let's get through it. Arteries, I already said it. Arteries always carry blood away from the heart, so away right there. This is also true when we deal. See, the away is always true. What is mostly true, arteries tend to have a lot of oxygen, but the connection, the exception to that is the pulmonary circulation and the umbilical arteries in the fetus. Those are the opposite. The umbilical artery is carrying blood away from the fetus to the mommy to get oxygen, the same way that the pulmonary arteries in an adult who's breathing air are carrying blood to the lungs to pick up oxygen. So although arteries are usually oxygenated and veins are usually deoxygenated, I wouldn't go depending on that and the color, because you might have to think about, the color blue for deoxygenated. You might confuse yourself if you don't remember those two exceptions. The one that has no exceptions, the one that is always true, is that arteries are carrying blood away from the heart. Anyway, moving on. Okay, here we have a pretty picture. And so we see the elastic arteries, the ones that have a lot of elastin protein. They are called conducting arteries. They're big channels that we pass huge amounts of blood through. Further down, we have the muscular arteries. Those are the ones that are going to do more vasoconstriction, vasodilation, and help send blood to different parts of the body. Understand that where we have an artery and a vein coming together, it's an anastomosis. Anytime you have two vessels coming together, it's an anastomosis. Venous anastomoses, where you have two different veins coming together, really common because they're easy to form because veins are at lower pressure than arteries. But we're getting ahead of ourselves. Let's move on. The other thing I want to point out here, we're going to be talking about the lymphatic system coming up. Why do we have lymphatic drawn in here at all? Because one of the main jobs of the lymphatic system, as we go through, we're going to talk about how this works, but when you're delivering blood from from arteries to capillaries to the tissues, and then when you do this, a lot of things like the albumin, like the red blood cells, like all the other plasma proteins, they tend to stay in the blood, stay in the capillary. But the fluid, the oxygen, the nutrients, they can leave and go into the tissues. Well, fluid goes out from the capillary beds at the arterial end, and at the venous end, the fluid comes back with the carbon dioxide and the waste in it, but not all of it. You never bring back as much as you put out. What that means is that you would have the tissues swell and get bigger and bigger and bigger and puff up with fluid, except that we have the lymphatic system here, which drains the tissues and returns the lymph, returns the fluid that came from the blood into the blood. So the lymphatic system is another method of getting the fluid back into the blood where it should be. The primary method, of course, is we return the fluid to the venules at the vein end of the capillary bed. But since that's not 100% effective, we need another method to drain the tissues, and the lymphatic system is that method. But we'll talk about that a lot more when we get to the lymphatic system. All right, so blood vessels. Blood vessels have a space in the middle of, think of them as a tube. The open middle part, that opening is the lumen. The wall around it has 3 layers. The tunica intima. Tunica just means layer. Think of a tunic, a cloak. Yeah, okay. So tunica intima is the middle layer that is in intimate contact with the blood. It's touching the blood. That tends to be smooth epithelial cells. We've talked about these a little more prior when we talked about blood clotting. Anyway, so you know that they're smooth, they're slippery. You know that they release chemicals that try to reduce clotting and prevent clotting. So tunica intima, touching the blood. And in a capillary, that's all you'd have. But in all the other vessels, you've got 3 layers. Then we have the tunica media that's around it. And obviously, media, it means middle. It's the middle layer. But the M should also remind you of muscle. This is where most of the smooth muscle is that squeezes the blood vessel or relaxes to let the blood vessel expand. So vasoconstriction, vasodilation, those are due mostly to the muscle found in the media, the tunica media. The tunica externa, this is going to be fibrous connective tissue. Think of like wrapping a tough, hefty bag around the vessel to keep it safe. It's flexible, but it's tough. It's hard to penetrate. It does a good job. Now, capillaries are just that endothelium, that tunica intima, okay? And the basal lamina they're talking about there, you've got some fibers of connective tissue to help hold the things together. And it's not on this slide, but it will be coming up. We might as well say it now. You've got 3 different types of capillaries. You have continuous ones, which are like an ultra thin tube or pipe. And then you have fenestrated ones. Der Fenster is German for the window, and that's where that came from. That's where that term came from. Fenestrated ones have little holes, little little windows all over, and that type of capillary is like a strainer or a colander that fluid passes easily through, but the holes are small enough it keeps a lot of other stuff behind. Keeps proteins and uh, keeps the proteins in the cells in the blood, but lets the fluid out. This is the kind of thing you find in the glomerulus on your kidney, where you have to do a lot of filtration. And then we also have sinusoidal capillaries that have big gaps where the cells don't quite come together. Anyway, so 3 types of capillaries: continuous ones, fenestrated ones, and sinusoidal ones. But the book will talk more about that. Let's move on. Okay, so tunica intima is the inner layer that's in intimate contact with the blood. It is just the endothelium, the simple squamous epithelium that lines all the blood vessels. Why squamous? Because it's flat. That means that it's thin. If it was a round cell and you had to get oxygen across it from this side to this side, that'd be a lot of work. But thin. Okay. So slick surface to reduce clotting, continuous with the endocardium of the heart. It's another name for the same layer. Okay, we've got, when we're in the heart, it's the endocardium. When we're in the vessel, it's endothelium. It's tunica intima. It's the innermost layer. Okay, and there's a little connective tissue basement membrane to hold it together. We only have this in vessels bigger than 1 millimeter. One smaller than that, we're just going to say, oh, it's a capillary. It's made out of just the endothelium with the basement membrane. So I'm not sure why they break it out there by size. But anyway, media, that's the middle layer, smooth muscle, and sheets of elastin. I'll tell you right now, you have lots of elastin in the elastic arteries, the ones that stretch real big. The other ones, the distributing arteries, less elastin, a lot more collagen to make them tougher. But these are the ones that do your vasoconstriction and your vasodilation. Vasoconstriction is reducing the size of the passageway that the blood can go through. Vasodilation is relaxing the muscle so it gets bigger. This is the main part. This is the biggest part of the wall, and it's what maintains blood flow and blood pressure. Yeah, obviously, blood pressure goes up. Blood pressure goes down because there's more room. All right, tunica externa, the outer wall. People also call the tunica adventitia. I won't be doing that. I'm going to use externa so you know it is external. So you know it's the outside layer of a blood vessel. It's mostly collagen fibers to protect the wall and anchor it in place in some cases. Says infiltrated with nerve fibers and lymphatic vessels. Large veins also have elastic fibers. Yeah, that's true. And here I mentioned this in passing when we were talking about coronary arteries providing oxygen to the heart because it has so much muscle, it can't just absorb it across the endothelium. Same thing. The vasa vasorum is the vessels of the vessel. The biggest blood vessels will have a bunch of little blood vessels that supply them with oxygen, supply their tunica media with oxygen. And they'll be located on the outside of the tunica media in this tunica externa. All right. So here we have the two. You can see, or I should say the three. We have an artery and we have a vein. You can see that they are both big vessels. And down here we have a capillary, very, very thin. You have nothing but endothelial cells and a basement layer. You don't have a tunica media outside that. You don't have a tunica externa outside that. In a capillary, the endothelial cells and the basement membrane holding them together is all you've got. But now look at the artery and the vein. Arteries tend to have thick muscular walls and a lot of elastin, a lot of elastic membrane. We have a much thinner wall in a vein and very little or no elastic. The elastin, we got two layers of elastin here. The elastin is a protein that has elastic properties. We got two layers of that in the artery because it's closer to the heart. It's at higher pressure and you need to be able to expand when the pressure comes through. Veins, you're past the capillaries, your pressure is very, very low. You don't need that. That's also why you tend to have the very thin muscle. In general, veins have big lumens and thin, flabby walls. And arteries have muscular walls with elastic properties. But there's this other difference down here. Notice the valve. Veins are full of valves, check valves, one-way valves that control the flow of blood. Arteries are not. Um, Arteries, the blood is coming at pressure from the heart, so that's what controls the direction it goes. In veins, it's very, very low pressure, and if you're going uphill against gravity, the gravity will pull the blood back down because you don't have enough pressure. So we have all these valves that will, as the blood tries to reverse and go downhill, slap, and that keeps the blood trapped until the next heartbeat when everything pushes upward and it catches the next little bit. Those valves are critical for things like moving from your big toe up your leg towards the heart. So the venous system is full of these floppy little valves. And the lymphatic system at even lower pressure, it has even more of these valves everywhere. All right, moving on. So here you can see what I said about arteries. There are different types. Elastic arteries, they tend to have a lot more elastin and not as much muscle, not as much collagen. I hadn't said that yet. Okay, so there were different types of arteries. We talked before right here about the ones closest to the heart. The big ones are elastic arteries, also called conducting arteries. They can stretch really well to help dampen the pressure spikes coming off the heart. The muscular arteries, the distributing arteries, are further away. And you probably guessed from their name, they got a lot of muscle. So now we have the cross-sectional picture so you can see it. Elastic arteries, big lumen holding a large amount, smaller muscle because they're not really going to do much vasoconstriction, vasodilation. They're mostly going to swell from the pressure and then return back to normal size, like a balloon would. The muscular arteries, they're the ones doing the vasoconstriction, vasodilation. So they got a whole lot of muscle. Okay. And then you look right here at the arteriole. That just means the little artery. And arterioles tend to have less of the muscle. They're still really important for that, but really important for vasoconstriction, vasodilation. But their whole wall is smaller because they're smaller vessels, they're thinner, they're getting closer to capillaries. You need to transition from this down to the capillary that's a single layer of endothelium. So arterioles are how we get there. They're the divisions that go off so we can get to capillaries. Anyway, let's move on. Oh, next one. Here we have the capillaries. And you can see they've got the endothelium. There's no basement membrane column here, but they have the endothelium. They don't have any elastic fibers. They don't have any muscle. They have no fibrous outside. I'm not saying the basement membrane doesn't have fibers. It does, but it's not like they have a tunica adventitia. Or I just said the word I said I wouldn't. They don't have a tunica externa that's full of those fibers. Now, veins and venules. Here's a venule. And a venule is like a little vein. It's like the arteriole was a little artery. But what's the difference? No elastic fibers at all. That's like not its job. And very little muscle. Compare that to an arteriole and you'll see how much more muscle the arteriole has. That's because we're controlling the flow into the capillary beds. The venules are mostly taking the blood that comes back from the capillaries, and they're not doing too much with it other than routing it where it should go. The vein, you can see that it has it's got the same small amount of endothelium. It has very little of the elastic tissue. It has some muscle. You can do some constriction and dilation, but it has a lot of the fibers around the outside. Basically, think of this as flabby, but tough so it doesn't tear easily, and full of valves to return blood. And part of this is, I should have made that more clear, part of this is valve too. Anyway, let's move on. So arteries divided into three groups. We got the elastic ones, the muscular ones, and the arterioles. You've already seen the pretty pictures. Let's do it. Elastic arteries, thick walls, low resistance lumen, meaning kind of big. The aorta and the major branches, they also call them conducting arteries because they conduct blood from the heart to the medium-sized vessels. That's lame. I'm probably going to be calling them elastic arteries because I want you to know that they stretch to dampen the pulses of pressure coming off the heart. You have the protein elastin found in all three layers, but mostly in the middle, the media, tunica media. They have a fair bit of smooth muscle, but that smooth muscle is not doing the vasoconstriction, vasodilation. Okay. They are pressure reservoirs that expand and recoil as blood is ejected from the heart. This allows continuous blood flow downstream, even between heartbeats. Great. Like I said, it dampens the pressure, it spreads out the charge. Elastic arteries will divide out into muscular arteries or distributing arteries. I'm probably going to call those muscular arteries to emphasize that they have very thick tunica medias full of muscle. Um, they could range from pinky sized to pencil lead sized. And these are most of the named arteries. They have the thickest tunica media, lots of smooth muscle, less elastin because they're not going to be swelling off that huge pressure. The tunica media is sandwiched between elastic membranes to compensate a little if you have to, but that's not its job. The ones closer to the heart, the elastic arteries, that's their job. And all these muscular arteries, they got lots of muscle. Why? For vasoconstriction, vasodilation. All right. Arterioles, they are the littlest of the arteries, the littlest of the things that contain all three tunics. Smaller arterioles are mostly a single layer of muscle surrounding the endothelium, so super thin, that close to being an artery. You lose that single layer of muscle, you lose the connective tissue fibers around the outside, and it's a capillary. All right, these control the flow of blood into the capillaries, and they do so. This is why we have the muscle layer, vasoconstriction, vasodilation. It's not mentioned here yet, um, but you will see it eventually. I want to point out that you also have a little circle, a little cuff. Well, let's use anatomy terms. A little sphincter of muscle around the entrance to the capillary bed, and you can open or close. Capillaries don't have blood flowing through them all the time. It's like the tissues there will get a spurt of blood, get the oxygen, and then they'll just wait until they're like, hey, we need more oxygen. And then you open up those sphincters again, send more blood. Then you shut it down again. We don't have enough blood in the body to fill all the blood vessels all the way, all the time. Some of the blood vessels have to get intermittent flow because we don't have that much blood. Anyway, moving on. Arterioles are also called resistance arteries because the change in diameter, we're talking vasoconstriction, vasodilation, will change the resistance to blood flow. And that makes perfect sense. If you have a big diameter for your vasodilate, it's easy to send the blood through. If it's vasoconstricted, it's a lot more work to send the blood through that narrow, tiny opening. Where's the picture? Yeah, we'll get to that. We'll get to that. I'm sure I saw. Okay, I did not see it. Hold on, we'll get back. In the second half, the PowerPoint that does the second half of this chapter, they have a really nice illustration where you're like drinking a chocolate milkshake and they give you two choices of straws, a thin, narrow straw and a large diameter straw. And obviously, the large diameter straw is easier to drink the thick milkshake through. Well, in the same way, that's why the vasodilated, thick, wide lumen is easier to pass blood through than the vasoconstricted, thin lumen, hard to pass it through. Let's see, where were we? We were, yeah, we were before that. Elastic arteries. Okay, yeah, we were on slide 17, and this is actually a really small, really short PowerPoint. Okay, so we did that. Capillaries are microscopic. They're tiny. Their diameter is so small that only a single red blood cell can go through at a time. We already said that. Some of them are so small that the red blood cell has to line up sideways, single file. The walls are nothing but tunica intima. You already know that. There is a little bit of basement membrane to hold the cells together, but nothing else. One cell forms the entire circumference. Parasites, spider-shaped stem cells, help stabilize the capillary walls, control permeability, play a role in vessel repair. Yeah, that's fine. And the capillaries supply almost every cell with blood and oxygen. There are exceptions. The cornea and the lens in the eye don't, because you can't have red blood in the way when you're trying to pass light through. And like cartilage is notorious for having poor blood flow. We talked about that in AP1. The poor blood flow is why if you get an infection in a joint capsule, it's a huge problem because you don't have blood going into the capsule to deliver white blood cells to fight. Anyway, moving on. So what are capillaries for? Gas exchange. What that means is they bring oxygen and nutrients to the tissues. They take carbon dioxide and waste away. They also deliver hormones. And they're the site where we're exchanging things between blood and the interstitial fluid. Interstitial fluid, if you remember, is just the fluid between the tissues. Oh, wow. It just dawned on me. I probably Alt tab. Yeah, that's better. You probably didn't see those two slides. I'm going to, yeah, we're still recording now. We do share screen. I backed up to where you were. All right, we are recording. So this was the next slide where I talked about the parasites, talked about what capillaries do. There we go. Now, this is where we talk about the three types of capillaries: the continuous, the fenestrated, and the sinusoidal. We're about to go through it, so you'll see. So continuous, all capillaries, endothelial cells have tight junctions with little gaps called clefts. Those clefts, those little gaps, are what allow fluid through the wall of the capillary, and small solutes. That means things like dissolved ions, not things as big as, say, proteins, not even small proteins. Three types of capillaries. You already know they're the continuous, the fenestrated, and the sinusoidal, and that's the order that we're about to go through them with. Continuous capillaries are, we have a lot of those in skin and muscle and lungs, in the central nervous system. The ones in the brain are a little different. We form a blood-brain barrier. That's tight junctions, no clefts. That's why we call it a barrier. So the blood there, oxygen, nutrients are passed through to the fluid, cerebrospinal fluid, and the cerebrospinal fluid supplies the brain tissues, the spinal cord tissues. Anyway, moving on. So here are continuous capillaries, and yeah, that's lovely. You can see that. Here are the fenestrated. I already told you, places where you have lots of filtration, like the kidneys. I didn't mention, should have, lots of absorption, like the intestines, or lots of hormone secretion. The short version is anytime you're passing a lot of stuff in, a lot of stuff out, you're going to use this type of capillary. It's porous like Swiss cheese. It has lots of holes that they call fenestrations. I already told you, German anatomists discovered this type of capillary, and der Fenster means the window. So it has a bunch of little windows. The fenestrations usually have, just like a window, usually has glass in it. Thin glycoprotein forms a diaphragm, so you don't just have everything passing through. All right, there we have our fenestrated. So you got that. Sinusoidal ones, you have fewer tight junctions. They can have fenestrations, but they have big clefts, and incomplete basement membranes. Lots of stuff can pass through, big stuff. That means we have them: liver, bone marrow, spleen, adrenal gland. Why would you do this? Well, big holes, you can load it with things. Say blood macrophages, the white blood cells, big things like that can pass through to get out and go and fight bacteria and nonsense we shouldn't have. The blood flow is kind of sluggish through these. That makes sense because you have changes in the molecules. You have to pass big molecules and things through. If you had fast blood flow and you have those big holes, sorry, these big holes, you'd be having a lot of things trying to get out quickly, and we don't want that. So since it's easy to get through the wall, slow flow through that capillary, we control the flow through the capillary to control what gets to the wall and passes through. All right, moving on. So capillary beds are a network of capillaries. They're what live between the arterioles and the venules. What we call microcirculation is the flow through a bed from the arteriole to the venule. The terminal arteriole, the last arteriole, will branch into 10 or 20 capillaries that are used for exchange of nutrients and gases. The capillaries will then drain into a post-capillary venule. A lot of times you have a shunt as well that we'll see that you use when you're skipping that capillary bed. Is there a pretty picture? Not here, but there we go. There's the pretty picture. All right. So we have our sphincters that control flow into the beds, and we have this shunt that you can go through. Yep, vascular shunt right there. So when you're not putting the blood through those capillary beds, you'll go through the shunt. And when you are, you will vasodilate these little precapillary sphincters, and then the blood will go out through the capillaries. Blah, blah, blah. All right. Flow through a capillary bed controlled by the diameter of the terminal arteriole and its upstream arterioles. In other words, we can vasoconstrict to make it hard for blood to get down to that capillary bed. Local chemical conditions and arterial vasculomotor nerves regulate the amount of blood. What they're talking about with local chemical conditions, if you've been running wind sprints and your legs are hurting, it's because you're building up lactic acid. That happens because you're not getting enough oxygen to those muscles. Well, guess what? That lactic acid buildup, it's a change in the chemical condition. Those are muscles screaming, "Give me oxygen!" And the response is arterial saying, "Oh, why, yes, here, let's vasodilate so lots of blood goes through." All right. Arterial and terminal arterial dilate when blood is needed, like we just said. They constrict to shunt blood away when it is not needed. There we have flow through typical capillary bed. Great. So in, say, intestinal mesenteries, we could have the vascular shunt, and we could have the precapillary sphincter. So it's not all capillary beds, but the ones in the around the intestine, vascular shunt will go arterial to venule, bypass the capillaries. You've seen the pretty picture right there. And you also have these little cuffs, these little rings of smooth muscle, the precapillary sphincter, that will control whether or not blood can even leave the shunt and go into the capillaries. Great. All right. So veins carry blood to the heart. That that's correct, accurate towards the heart. A lot of times, since we have that nice mnemonic, arteries carry blood away, I'll say veins carry blood very close to the heart. Um, the VV and AA then works. Uh, Veins form when capillary beds unite in venules, and the venules merge into the larger veins. Um, Venules have endothelium and some parasites. They're really porous, so blood and white blood cells, fluid and white blood cells can go into the tissues. Larger venules are going to have a couple really thin layers of smooth muscle cells. We form veins when the venules converge. Veins have all three layers: the tunica intima, the tunica media, the tunica externa. You need to know those names, by the way. But they have really thin, flabby walls. If you compare them to arteries, arteries are kind of thick, muscular walls. Veins are kind of thin, flabby walls. Why? Arteries are closer to the pump. Arteries are closer to the heart. Arteries are exposed to higher pressures. Veins further away from the heart, past the capillaries. You got very, very little pressure left. So you don't need to build this thick, massive wall. And the tunica media is thin. The tunica externa is thick, so lots of collagen to hold things together. Large lumen, thin walls. That means you can have lots of blood stored in them. That's why I call them capacitance vessels or blood reservoirs. More than half of the blood is in the capillaries. 65% of the blood is in the capillaries. Why am I saying capillaries? 65% of the blood in the body is in the veins. 65% in the veins. All right, moving on. Actually, I know why I said that. Because C in capacitance got me Anyway, so here we have the difference in an artery and a vein. Okay. So if you look at the artery and the vein, you see this light purple staining, that's where your tunica externa is with all your collagen fibers. We've still got that going on. But look at in the artery, we see this dark staining tunica media, and we've still got one here, but it's really thin. And here inside that, we have the endothelium, the tunica intima. We still got that, but it's so thin you're not even seeing it. So vein, thick, flabby wall. Artery, thick, muscular wall. Great. So how much blood is in each type of vessel? If we look at this, 60% plus, the last slide said 65% of the blood is in the systemic veins and venules. Only 5% is in the capillaries. Remember, I said most of the capillaries are closed most of the time. This would be different if we were opening all the capillaries up, but we don't have enough blood to do that. We'd go into hypovolumetric shock if we tried. 8% of the blood in the heart. The pulmonary vessels, of course, are the ones that go to the lungs. Pulmonary blood, that's 12%. Systemic arteries and arterials, 65%. So you can see the lion's share of the blood is in the veins, in the venules. All right, moving on. Veins have lower pressure than arteries. You've heard that. So that adaptation ensures the blood will return blood to the heart. Large lumens, very little resistance. Also, tons of these venous valves to prevent backflow. If you're going uphill against gravity and then between heartbeats, the blood wants to come back down. Sorry. Uphill against gravity, and then the blood wants to come back downhill. Those valves will fold in from the drop in from the sides, close the lumen of the vein so the blood can't go down. Okay. So yeah, these venous valves really common in legs and arms to get blood back from the extremities, from the fingers, from the toes. Uh, Veins also have these venous sinuses. We already talked about one when we did the heart, the coronary sinus on the back of the heart. And you'll see as we read these 3 bullet points, you'll be like, yeah, that's it. Flattened veins, extremely thin walls, only endothelium, there is the example, coronary sinus of the heart, dural sinuses in the brain. All right. Hey, guess what? We've done this. They're showing it again. We've talked through this, you already know. We've done that. We've done that. Great. Homeostatic imbalance. Varicose veins. You've seen them. They're the blue-looking tracing of veins that people with pale skin have. You can see the bluish veins through it, especially in the legs. Dilated and painful veins due to incompetent or leaky valves. If you are on your feet all the time, if you are standing all the time, if you're obese or pregnant, the blood's going to pool in the lower limbs. It's going to weaken those valves, and the vein, because it has thin walls, actually start to bulge. That's what lets you see it, is that at the bottom it's bulging, and thus getting closer to the surface of the skin. Anyway, 15% of adults have varicose veins. What can cause these varicose veins? Uh, Elevated venous pressure, high blood pressure, straining to deliver a baby, straining to have a bowel movement. This raises intra-abdominal pressure. The bowel movement thing, if people strain, you can get varicose veins of the anus. That's what a hemorrhoid is. What in common speak people call piles. It's hemorrhoids of the anus. of anal arteries. All right, so an anastomosis, I already told you, it's where vessels are coming together. The veins tend to have a lot more anastomoses than the arteries, simply because they're at lower pressure. It's easier to form them. So, arterial anastomoses are alternate paths to deliver blood flow, even if one artery is blocked. Where it says here, common in joints, abdominal arteries, the brain, and heart. We don't have these in the retina, the kidneys, or the spleen. The big one in the brain that you're going to have to learn is the circle of Willis, which gives you, I can safely say multiple, but it would be better to say 3 or 3.5 ways to deliver. I say and a half because the two vertebral arteries fuse to become one basilar that goes up to the circle. But it gives you multiple ways to deliver blood to the brain. So if you block one, you're not cutting that part of the brain off from blood. You have blood coming up here, and because of the circle, it still goes to the back if you've blocked your basilar. Anyway, um, we also have arteriovenous anastomoses. We talked about the shunt already. That's a good example. Venous anastomoses. There are tons of these because low pressure, easy to hook the veins together without causing a problem. And so that's lots of ways to drain the blood from the organ. Hey, and that is the end of this PowerPoint. So let's shut this one off and upload it. Oh, come on. All right, there we go.